origin of antibiotic resistant gene and multiple cloning site. This is artificial gene first part. Example PBR322. First widely used artificial plasmid PUC18, PUC19, high copy number used for blue-white screening process. So they're based on the for structurally studies thoroughly in the plasmid, nature plasmids, that nature base, that character feature base, our biotechnologist prepared the artificial plasmids. The first one, PBR322, the second one, expression vector purpose. Cloning purpose, example, PBR322. Gene expression purpose, that is called a PET series examples. To express the foreign gene as a protein, features, promoter, ribosome, binding site, operator, a terminator, and selectable markers. These are all features are based our biotechnologist. These are prepares the expression vectors purpose PET series. So each E. coli PGEX used for the expression fission protein with a GST tag. This is the second type of the artificial chromosomes, artificial DNA. But next one. So this is third one. Third one is the reporter vector purpose to study the gene expression regulation. Report the gene like a GFP luciferase, also called a beta galactosidase. These two type of the vectors are artificial vectors. Artificial vector plasmids. It contains a luciferase enzymes, also got gain fluorescent protein. It is also PE. GFP plasmids. Then fourth one also shuttle vector purpose to replicate the different host species such as bacteria and yeast. Then bacteria and yeast mammalian cells. The two origin of the replication and selectable markers for the host. Example YEP, yeast episomal plasmids. Work in both E. coli and yeast. This is also one type of the additional report of the artificial chromosome. Then RNA interface, also got a gene knockdown vectors. The purpose to silence of the specific genes. Gene expression, gene regulation, gene cloning, and silencing. That four characters purposes based to our biotechnologist to prepare this type of the artificial plasmids. Features shRNA or siRNA, SA means silencing, expression encapsulates. Example, PSUPR used to express short hairpin RNAs. Then sixth one, viral plasmid vector. Viral, that purpose is they use the generator viral particle for high efficiency gene delivery. High efficiency gene deliveries, gene transfer formation, then lentiviral, retroviral, adenoviral, AA viral. These are all is also examples of the then viral plasmid vector. Then seventh one, transpose-based vector. It can purpose to insect genes randomly or specific sites using transpose activity. Randomly genes are transferred to the plant cells. That is called transpose-based vectors. Features transpose elements flanking the genes of insect or interest. Eighth one, gateway vectors. Purpose for easy to efficiency recombination based on the cloning. The recombinated sites which is also inserted to the traditional also restriction sites. This is also one example of. Then also got a replication of the plasmids. What are the important of the replication? The plasmid replicate autonomously because they have their replication origin. The enzymes are involved in a plasmid replication are normally cell enzymes, means DNA, also got RNA enzymes, particularly in case of the small plasmids, which are multiply into the two based on the semi-conservative process. but some large plasmids carry genes that code for enzymes that are specific for plasmid replication. The plasmid passes relatively few genes, generally fewer than 30, and the genes are concerned primarily with a control of the replications. So the genetic information carried in the plasmid genes is not essential to the host because the bacteria that lock them usually functionally normally. That is one character. It is also got then plasmid curing. Then what is a plasmid? The plasmid can be eliminated from bacterial cells and it's a process called a curing. Eliminated function. Based on the eliminated function, that is called a curing. Curing may be take a place spontaneously or it may be induced by various treatments which inhibited plasmid replication but do not affect bacterial chromosomes. So then only plasmid is curing. Curing means that quality is rises. Then that quality rises do not affect of the original chromosome of the nuclei of bacteria. The chromosome replication and cell reproduction, the inhibition plasmids are slowly diluted out of the growing bacterial populations. Some commonly used the curing treatment agents acrylic dyes, ultraviolet rays. Dyes. Dyes means here some type of the coloring substance which can be cure the plasmid activity or activated the plasmid activity. Plasmids also called UV rays and ionized radiation, thiamine starvation, and growth above optimal temperatures. It can base to the plasmid curing. These curing treatments are agents in interference with the plasmid replications of bacterial chromosome. This is also one plasmid curing. Use of plasmids as a cloning vector. What are the use? The plasmid which is a small size, which makes the plasmid easy to isolate and maintain. That is one quality. Independence of origin. The second quality. Multiple copy number within a short period, large number. That is also third quality. The fourth one, presence of selectable markers such as antibiotic resistant genes, which makes a detection and selection of the plasmid containing clones. Antibacterias. Also genes are insertion to the plasmid vector that can be easy to the gene cloning process, which are major important user for the plasmid as gene vectors. So this is also control of the CRISPR plasmids. It is also artificial type of the genes. It is one important one important. But here is a plasmid vectors. Here is also what are the role of the biotechnology. What are the role of biotechnology in plasmids? The role of plasmid in biotechnology. How many functions are there? The majorly biotechnology purpose, 11 functions are maintaining through the plasmids. First one, gene cloning. What is the role of plasmid? Use as a cloning vector or inserted and replicate foreign DNA. That is example, plasmids like PBR322, PUC19. Second one, recombination. Recombinant DNA technology means carry genes from different organisms and help in creating recombinant organisms. Carry genes, that is also first gene cloning, recombinant DNA, just differentiation. Carry genes from the different organisms and help the creating recombinant organism. Also got GMOs, transgenic bacteria. The third important purpose of the bacteria application or biotechnological application is production of the therapeutical proteins. So expression of foreign genes coding for useful proteins. Example, human insulin, growth hormones, productions. It is a one character. Then the fourth one, gene expression studies. Expression, plasmids carry promoters and genes to study protein function. Study of a gene regulation and protein function. Then the fifth one is agriculture biotechnology. The plasmid is a more important role in agriculture biotechnology. That is got tumor-induced plasmid, usually genetically modified plants, agrobacterial-mediated gene transformation here. Development of paddy, cotton, pest-resistant crops. Then also sixth one, vaccine development, delivery antigen coding genes of DNA vaccine developing COVID-19 resistant. So before 3 years, our Telangana biotech institution, which can base to the COVID-19 quality of vaccine releases, that is called vaccine development, depends on the knowledge of biotechnology. That also sixth one. The seventh one, gene therapy, used to deliver therapeutic genes into patient cells. The treatment for genetic disorder, cancer. The cancer diseases are also curing, which help out the plasmid vectors. The eighth one, reporter gene assay. Plasmids with reporter genes help monitor gene activity. Use of GFP, green fluorescent proteins. Green fluorescent proteins productions. So then the eighth one, reporter gene assays. Plasmids which reporter genes help monitor gene activity. Then also ninth one, biosensor or bioremediation. Engineering or plasmid help microbes are detected creates. Also got a oil spills. Also clipping using modified pseudomonas. 10th one, study of antibiotic resistancy or plasmid carries a resistancy genes used the microbial. Then last one, horizontal gene transfer help study conjugation and a plasmid transformation between bacteria. Understanding bacterial evolution and ecology. These are out of 11 important rules in plasmids for biotechnology. This is also last one. What are the differentiation of the how many plasmids? What are the character features of the plasmids? Then it is how many features are there? Major 10 features to euplasmid, R plasmid, cold plasmid, degradable plasmids, triplasmids. It's so 5 plasmids are differentiation here. Then full name. Then also got a fatally plasmids, testally plasmid. So cholecinogen, genic plasmid, degradative plasmid too. That are found in E. coli, bacteria, Pseudomonas bacteria, agrobacterium. Then main function is enable, provides, then produce. Then conjugative additive is large recombinant, large varies, but transfer mechanism has a 6 pillars. Then conjugation. So these are gene content, role gene transfer, significance in medicine, effect of the cause, effect of the host, also unique features. These are all is also measure to classify the and 5 type of the plasmids variations or differentiations. Last one is also filling the blanks. These filling the blanks is a major cloning plasmids dashes are used for primary to dash DNA fragments. Clone. A typical cloning plasmid contains a origin of replica, antibiotic resistant gene and multiple cloning sites. Then the abbreviation for a multiple clone site is called MCS, multiple clone sub sites. Then the origin of the replication of essential for dash replica. The expression plasmids are used for dash expression, gene expression. And a expression plasmid must contain a dash promoter gene. Then the gene of interest in an expression plasmids is typically followed by a sequence. This is also terminator, but dash is also enhancer elements, so can be added to the expression. A plasmid uses to reduce the gene expression known as dash gene knockdown plasmids. Gene knockdown. Then gene knockdown is often achieved using short hairpin. Short hairpin. The abbreviation of short hairpin is called shRNA. Then also this, but here viral plasmids are used to the delivery genes through viral. Then lentiviral, retroviral, and viral plasmids are examples of viral. Then also then retroviral plasmids are integrated into the host genome. Expression vectors can be used into the both bacteria and eukaryotes. A strong promoter is also high. That is also, but here plasmids can be introduced into the host cell through transformation. So in expression vector, transcription is followed by translation. Cloning vector log the necessary elements or genes is called expression. Knock down the plasmids, reduce dash expression. Then a transcription terminator stops a transcription. Viral plasmids are often used in dash gene therapy. Then multiple cloning sets contains a several unique restriction enzymes. Then the genes of the interest is inserted into the dash, means MCS. Then antibiotic selection ensured the transformer cells. This is, then viral vectors are allow for dash, means also efficient. Reporter genes are provide a visible array and measurements in output. Then enhance increase the rate of the gene is also called transcription. Then expression of the plasmid can be used to produce a recombinant protein. Then plasmids are commonly used in a genetic engineering. Then the origin of the replication allow the plasmids to be copied the host. These are important of the all type of the importance of the multiple or filling the place. Okay, sir. Thank you very much.
Thank you very much, sir. UGANDHAR, sir. Today, most important topic related to the extranuclear genome or the prokaryotic genome, especially the cytoplasmic inheritance or the plasmid DNA or extrachromosomal DNA. The best example, one of the best example in prokaryotes, we are observed the plasmid DNA, which are used as a vector, for the genetic engineering process, the gene transfer, the biotechnology. So types of the vectors and types of the synthetic vectors or the artificial vectors. Sir has clearly explained what are the importance of the vectors and also the most important bits related to the vectors, plasmids. Sir has clearly explained, and the importance related to biotechnology, Sir has clearly explained. At the same time, the what are the main characteristic features of the plasmid, Sir has clearly explained in this topic. So, my dear friends, if you have any doubt regarding of this topic, please ask our respected resource person, Dr. THIRUNAHARI UGANDHAR, sir. Is the most eminent person in the field of plant biotechnology and tissue culture and also the molecular biology. So, excellent presentation, and it is very crystal clear to understand, to listen carefully. It is very useful. This video link is available in YouTube channel also. So thank you very much, sir, for your wonderful presentation. So with your permission, we're going to wind up the session. Thank you, sir.